I'm Allison Hahn, Field Application Scientist with Illumina. In this session, we will explore the Illumina sequencing workflow, and I will introduce sequencing by synthesis, Illumina's sequencing technology. Here's a high level of the Illumina sequencing workflow. In the first step, we extract our genomic DNA from our sample. During the library preparation process, we convert our DNA into a sequenceable form. Next, we complete cluster generation, where we apply our library to a flow cell. The sequencing then occurs inside a flow cell on one of our sequencers. Finally, during data analysis, we determine what the DNA sequence was of our original extracted genomic DNA. In order for sequencing to be possible, we must convert our DNA into a library. The library is made up of 4 major parts. The first is our DNA insert. This is the DNA that we want to sequence from our sample. Next, we have our sequencing primer binding sites. These allow for the sequencing reaction to be initiated inside the sequencer. Then we have our indexes. These are unique barcode sequences that we apply to each of our libraries that will allow us to mix many samples together during a single sequencing run. And finally, in order for cluster generation to occur, we need the P5 and P7 regions on either ends of our libraries. These parts in colors on the outside of our DNA insert is what we refer to as adapters. Let's talk a little bit more about the library prep workflow. When we extract genomic DNA, it is composed of long fragments of DNA or chromosomes. The Illumina sequencers can only sequence short fragments at a time, so we must fragment the DNA. We can do this through either physical or enzymatic process to convert the long stretches of DNA into short pieces. Next, we add our adapter regions to the ends of our inserts, and then we PCR amplify up the libraries. After library prep, we move the sample to the sequencer to complete cluster generation and sequencing. Cluster generation is done to fix the library to the surface of the flow cell using PCR that will copy the DNA in place. Next, we add fluorescently labeled nucleotides that have a block on the end, so only one base is added at a time by the DNA polymerase. We take pictures at each cycle as we add the nucleotides, and we're able to determine which nucleotide was added and therefore can determine the sequence of our DNA inside of our library. Now let's talk about sequencing by synthesis, or SBS, in much more detail. First, this slide shows our empty flow cell. The surface of the flow cell is covered with DNA primers or oligos that are complementary to the P5 and P7 ends of our libraries. These oligos will bind to our libraries once we add them to the flow cell. At the beginning of cluster generation, we add our libraries to the flow cell. You see here our library, which has the purple on one side and the blue on the other side. These are the adapters that we added during library prep, and the gray in the middle is our DNA insert that we're going to sequence. Our library is now fixed to the surface of the flow cell via interactions on the P5 end of our library. Note, during cluster generation, we're going to make thousands of copies of this one strand, but to keep it clear, we're only going to show a single strand during this session. To start our sequencing reaction, we add one of our sequencing primers that binds to the adapter regions at the top of our library. We add all 4 of our nucleotides, which Illumina creates by adding a unique fluorescent label to each one of the nucleotides. The DNA polymerase will now know which of the 4 bases to add. In this example, the first base added is a red A. We take a picture and we're able to determine that at this library, an A base has been added. The next step of the cycle, we remove the fluorophore and remove the 3' block. Now the A base that we sequenced in the first cycle looks like any other nucleotide. And now we're going to add all of our 4 labeled nucleotides back to the flow cell They will flow over, and the DNA polymerase will determine which base to add next. For this library, a C or green base has been added. And we're going to continue to deactivate and remove the fluorophore from our nucleotide. And we're going to do this one base at a time. And each cycle, we're going to add all 4 nucleotides, and the correct one will get incorporated by the DNA polymerase. In this cycle, a green C has been added. To complete the cycle, we remove the label, and then we start another cycle, and we sequence the next base. This base added is a blue T, and then it's removed. We continue adding the nucleotides. A green C is added and removed. And finally, we reach the end of our DNA insert and have incorporated a yellow G, and then it's removed. So, what was the sequence of our DNA insert for this library? Answer: looking back at which color nucleotide was added for each cycle, we can determine the sequence of this library. The answer for this library, the sequence is A, C, C, T, C, G. So now let's go back to our flow cell and look at all of the primers again. Each one will have a library that we can sequence in the same way at the same time. So now when we add our libraries during cluster generation, we have 3 different libraries that will flow through and bind to these specific oligos. Remember, this is happening all over the surface of the flow cell, not just at this 3 location. So now when we go in and we add our primers and start the sequencing reaction, we add all 4 of our nucleotides. Again, they have the fluorescent label on there, and the polymerase knows which of them to incorporate. So as it's flowing through, these 3 libraries will have the correct base added to the string. In this example, library 1 and 3 are yellow and library 2 is green. So I want you to go back and write down the bases for these 3 libraries. So as we go through, this is for cycle 1. So again, all of the nucleotides have been added at the same time, and the block has prevented any more being added. So all 3 of our libraries will have the nucleotides added to them by the DNA polymerase. And the important thing to remember is that these 3 libraries will always appear in the same location in each of our pictures. So as we continue for each cycle, adding and removing our fluorescent nucleotides, we're going to continue this process and build the sequence for each of our individual libraries. Okay. Write down the 3 bases that have been incorporated for this cycle. We're continuing now with cycle 3. Which 3 nucleotides have been added to our libraries? And again, we're just going to continue this process of sequencing by synthesis, one base at a time. At some point, some of our libraries may run out of DNA to sequence. You can see library 3 here is shorter, and so it is now gone dark in our images, but library 1 and library 2 will continue to be sequenced. And again, library 1 and 2, write down the base. And now you can see library 1 has gone dark, but library 2 continues for the next 1 cycle and 2 cycles. So now we can go back to all of our images and determine what the sequences were for each of our individual libraries. So what are our sequences? These are the answers for library 1. It's G, A, T, G. Library 2, C, T, G, T, C, G, A, T. Library 3, G, C, T, C. So now that we've collected the data from our sequencing run, we can look back and determine which bacteria did our libraries come from. We do this by comparing our sequences from the sequencing run to the known reference genome of E. coli and STEC. So our 3 libraries are shown here, and if we look down at the E. coli genome, if we look at the E. coli genome listed here on the top, as well as the STEC genome, also listed on the top, you can see that the difference is in the STEC genome, the third base is a G, whereas in E. coli, the base is an A. We take all three of our libraries and we compare the sequences that we've collected to these two references. And what we find is that library 2 and library 3 are found in both genomes, but library 1 is only found in an E. coli. This tells us that the DNA that we sequenced is from E. coli and not from STEC. The goal of this experiment is to determine if our libraries came from E. coli or STEC DNA. Once we know this, we can determine which sample is positive for the STEC contamination and notify the CDC that this sample is making people sick. So we did this for our 3 libraries, but you have to remember that really on the surface of the flow cell, this is happening millions of times all over the surface. And so we're able to collect millions of library reads for our samples at the same time. To see this process in action, please click on the link for the SBS video on YouTube. If you have any additional questions, please post to the Illumina Educators Workshop page on Workplace.